Welcome everybody. This is Julian from Straight from a Scientist. I'm here with a special guest, Georgia from Australia. We have quite the time difference. It's dinner time in Australia right now, and I'm coming home for lunch right now. So thank you for coming on, Georgia. How are you doing tonight?
I'm good. Thanks so much for having me here.
So tell us a little bit about what you are studying. You're getting your PhD in biochemistry and immunology, correct?
Yes, that's correct. So yeah, my PhD is in biochem and immunology, but in particular, we focus on cell death research, and in particular, look at apoptotic cell death, so a programmed form of cell death, and basically look at what happens after cells die through this process and the consequence of this in both homeostatic and disease settings.
Interesting. Now, you were telling me before that you're focusing on influenza infections, as far
Yeah,
as the
yeah.
disease states.
Yeah, so when we get infected with the flu or influenza, this causes all of the immune cells to infiltrate into the lung, and therefore they are all exposed to the influenza virus and all undergo a severe amount of cell death. So through apoptosis and pro-inflammatory cell death as necrosis. So yeah, there's a really heightened level of cell death. So understanding the process of this is really important to looking into disease severity.
Interesting. Well, for our listeners that are not familiar with these terms, can you explain a little bit what apoptosis is, and of course its difference between apoptosis and necrosis?
Yeah, so cells can die in lots of different ways. The most common way is, or most researched way, is apoptosis, and this is a programmed form of cell death. So they follow a series of sequential steps going through this cell death pathway, and it's generally a anti-inflammatory process. So it doesn't really cause any severe effects to the surrounding in the tissue or body. And this is in contrast to some other types of cell death, namely necrosis. And these are generally not as well regulated or controlled and are actually quite pro-inflammatory, so they can cause inflammation and therefore are quite harmful to the surrounding cells and tissues.
That's really interesting. So, I've heard apoptosis described as kind of like a cell suicide. Is that correct? It's kind of like
Yeah,
the body. So
definitely.
the body is telling the cell to die. It said, "We don't need you anymore, or you're infected, or you're not good for us. Please die." More or
Yeah,
less.
that's exactly what happens, and especially in the case of my research in influenza. So if the body realizes that these cells are getting infected with the virus, they want to try and minimize any ability that the virus has to replicate and spread. And
Exactly.
as these viruses are using the host cell machinery to aid their viral replication, they shut down that host machinery and commit suicide. So it can limit that viral spread.
Wow, that's really cool. So, the average person would think cell death, this is not good, But in this case, the cell death is actually protecting the host from severe spreading of the disease or of the virus in this case.
Yeah, well, that's what we think. But I guess at the moment, we're not really too sure what's going on. A lot of the time, cell death is definitely important to remove harmful cells and old cells. But in the case of diseases such as influenza, whether this is a good or a bad thing and what type of cell death is good or bad is not really known. So that's something that my research is trying to understand.
Okay. So one thing that I don't understand very clearly is that what happens after the cells die. So you said you're studying specifically the clearance mechanisms. How are these, or I guess the dead cell tissue, how is it cleared out of the body? What is the mechanism of that?
So after a cell undergoes apoptosis, so that programmed form of cell death, it can then fragment into small vesicles called apoptotic bodies. And these have been suggested to aid this clearance process. So a way to think about that is basically when you eat a steak. So when you eat a steak, you use a knife and fork and cut it up into smaller pieces, and then it's easy for you to chew and eat and etc. And that's exactly what people think happens with cells. They break down into these smaller vesicles. So then the surrounding cells, known as phagocytes, can come around and eat them up in these small bite-sized pieces.
Okay. So they get chopped up like a steak and then kind of get cleared out of the cell. Interesting.
Yeah, that's exactly super easy.
And where do they go after that, though? How are they excreted through the body, or do they go into the bloodstream, or?
Yes, so the surrounding phagocytes will engulf them and then break them down themselves, so just completely degrade them.
Okay, that's really cool. Interesting. So we talked a little bit. You said that influenza is one of the disease states that you're studying. Are there any other disease states that are related to apoptotic cells and their clearance?
Yeah, definitely. So in our lab, we focus on a wide variety of disease settings. So obviously, as mentioned, I work on influenza, but there are people in our lab who focus on cardiovascular disease. We also have a cancer model, and another really good one is an autoimmune disease model. So many autoimmune diseases are characterized by having a large amount of cell death. So again, understanding the role of this cell death and the consequence of that fragmentation into these small vesicles is really important for that disease pathology.
Yeah, especially autoimmune. I was going to say, I guess autoimmune could, they're telling the cells to die. Maybe you want to decrease the apoptosis in this case, right? In an autoimmune case.
Yeah, possibly. And especially if they're breaking down into those vesicles, which could contain potentially harmful components. What we're also looking at in our lab is a series of pharmaceutical compounds which can manipulate that fragmentation a step. So if we can find that these vesicles that form after cell death actually contain harmful components, we could potentially block that fragmentation event to then promote efficient clearance and removal of those harmful components.
That's crazy. That's huge. Wow, that's really cool. Yeah,
Does that make sense?
Yeah, it makes a lot of sense. Coming from a neuroscience perspective, though, I wanted to ask you about some neurodegenerative diseases. Because in a lot of the main neurodegenerative diseases like Alzheimer's, Parkinson's, there's always an accumulation of protein in the brain. Now, do you think this has anything to do with cell death that has not been cleared properly? Or
possibly, and it's actually quite interesting to talk about that kind of protein aggregates in terms of not only cell death but those extracellular vesicles that I've been talking about, because quite a lot of the field in the EV field, the extracellular vesicle field, look at the localization of molecules, including these protein aggregates found in those disease settings in those extracellular vesicles, and I've looked at them to be able to be trafficked from one cell to another. So it's definitely an appropriate field to further our understanding.
yeah, there's some therapeutical approaches. They're trying to produce clearance mechanisms for these protein aggregates and see if that helps the disease state, but they're still working on that. That's interesting.
Yeah.
Yeah, like the main ones are tau and amyloid beta. I'm sure you have heard of those.
Yeah, sure. Yeah.
Interesting. Cool. So I wanted to ask you about autophagy. How does autophagy play in what role does it play in I can't say this word. Apoptosis. I'm just gonna say apoptosis.
Yeah, apoptosis. So autophagy is also related to another form of cell death. So as we mentioned before, we have apoptosis and necrosis, and autophagy can also be related to another form of cell death, which is really cool. And normally in normal cells in a resting state, they undergo autophagy or autophagy, depending on how people want to say it.
I've heard it's said different.
Yeah, autophagy as well. But that's basically where it's the cell's recycling systems. They create these membrane compartments inside the cell to then break down the components within the cell. However, this can also be used as another cell suicide mechanism to degrade the entire cell as another form of cell death and break it down and remove it.
Interesting. And this can be positive or negative, I guess, depending on the circumstances.
Yeah, definitely depending on the circumstances. Most of the time it's a positive thing. It's another defense mechanism like apoptosis to remove these harmful cells. If they detect a pathogen or possibly a cancerous cell or something like that, they can induce this positive form of cell death to remove it away from the body.
That's really cool. Okay. So explain to us a little bit about how you got into this field. It's a very specific field, but tell me a little bit about your pathway into science.
Yeah, I guess so I did my high school in a small country town, and then I moved to Melbourne, which I'm in now, to study my bachelor in biotechnology and cell biology. And I guess I went through all of my undergraduate years not exactly knowing what I wanted to do. I only really knew that is I liked science, and that was about it. And then I did some work experience throughout my undergrad year, and then I got into honours. So I'm not sure if you have an honours program in Spain. Do you have that?
I guess for undergraduate, you're saying.
Yeah. So, but
Yeah, we have honors.
I
Yeah,
guess,
I did my undergraduate in the U.S. I'm just doing my master degree here in Spain.
okay.
Yeah.
Yeah. So I did my honours, which is technically undergrad, but kind of seen as postgrad studies here. And then after my honours, I just continued on with the same research group that I was in to do a PhD. And I guess I was quite fortunate because my honours supervisor, I was his first student, and he was really just starting out on this new bandwagon of this apoptotic cell disassembly process. So I was in a really good position to initiate the team and the lab with him and set up some really cool projects, and just really fell in love with the research and the project.
That's really cool. That's really cool. So, and what are your plans for when you finish? You said you're in your last year of your PhD, so congratulations.
I am.
You're almost there.
I know, counting down the months. So yeah, hopefully I finish up in the next 3 to 4 months. That's the plan, and then I'll definitely stick around in research for a while. I think it'd be really good to try to apply for a fellowship in the next few years and see where that takes me. But I also really enjoy communications, so I'm definitely keeping my eye open for more scientific communication roles throughout Australia to see if that is a possible career pathway as well,
Cool.
in addition to research.
Yeah, well, if you need to ask us any questions, we have a lot of contacts, and we're always growing, so we can try to help you. But maybe not in Australia. You might have to come to the U.S.
I'll definitely keep that in mind.
Cool. Thank you. So let's see. I wanted to something that I've been researching a lot recently, just for my own personal, is fasting, and I have read a lot about fasting, how it promotes apoptosis and autophagy or autophagy.
Yeah, that's the one.
Have you heard anything about that? Have you?
I haven't, but that's a really interesting thing to read about. I'm not entirely sure how that would work. What did you read about that process?
Well, I'm not an expert at any. I think from my understanding was that fasting can sometimes, don't quote me on this, can eliminate like pre-cancerous cells through apoptosis.
Okay.
But you'd have to read more up on this. I'm not an expert. I haven't done my reading. So just
Yeah, but
things
I
that
guess
I've noticed and read. I don't know if you've heard it. I was just asking you because
it definitely highlights a really important thing that everyone's realizing these days, of how much the environment is really impacting on what's happening inside us. So
exactly.
the diets is becoming a huge thing at the moment in not only cell death, but that extracellular vesicles field that I was speaking about, and people are really realizing how much it really contributes to regulating our cells at the molecular level. So it's a really hot topic to read into. So I might look into that paper.
Yeah, look into that and let me know, and maybe I'll send you some papers that I have looked at recently.
Yeah, for sure.
Yeah, it seems to be. I like this though. I think it's good. I think the food as medicine movement is huge, and I think it's important. I think people need to take a more holistic approach on their health, instead of just going to the doctor looking for a quick cure.
Yeah, definitely agree.
But yeah, well, okay, cool. I'll send you some of those. Let's see. Are there any things that affect this clearance of dead cells from your body, like metabolism, maybe, or I don't know.
I guess not necessarily those environmental factors that we've been speaking about, but a lot of the time in different disease settings. So because in some diseases, they can promote different pathways of cell death that they may impair that clearance process. So, in particular, one of the most commonly studied characteristics is that if these apoptotic cells, which if you remember are normally anti-inflammatory, if they're not quickly cleared by these surrounding phagocytes, that then they can actually rupture and explode and release all these harmful pro-inflammatory molecules. So in lots of disease settings, like in those autoimmune diseases, that's really important to understand and to really optimize a system where we can enhance the clearance and removal of those apoptotic cells before they get to that rupture and pro-inflammatory state.
Yeah, wow, okay. Interesting. Yeah, what about I know one of the main, in neuroscience, the main autoimmune disorder is multiple sclerosis. Do you know anything about that and its relation to apoptosis, or it has to do
Not
with
specifically
death?
that one.
No,
Yeah.
that's fine.
Where
I
the
won't ask you any more trick questions.
That's okay. The model that we are going to set up is in SLE, which is
Okay. Yeah, that's a
yeah,
good
it's
one.
similar. Yeah, so maybe ask me about that in a year's time after we've got some good results, and I can
Okay,
share that with
perfect.
you.
That's really cool. Awesome. So you live in Australia. I have to ask you, what do you do outside of the lab? You're surfing. When I think Australia, I think of the beach, the Great Barrier Reef, kangaroos,
Yeah, well,
dangerous
all that
animals.
is that's all north. So I'm down in Melbourne, which is down. So it's generally pretty chilly in Melbourne, especially now. It's in what month is it? May, so it's getting pretty cold. But outside the lab, I really enjoy the outdoors. So I grew up on a small farm in the country, so I go home there quite often, and really enjoy going out there and going camping and fishing and all that kind of stuff. Really getting amongst it in the Australian wildlife,
The outback,
seeing lots
right?
of kangaroos.
I was gonna
Yeah.
ask you so for the country, for people that don't know, I guess in Australia the country is the middle of the island or the continent.
Oh, it's just kind of everywhere. So if you
Everywhere's the
drive,
country except for the
yeah,
big cities.
yeah. So if
Okay.
you drive basically 2 hours out of the city in any direction, you're in the country now.
Okay, so that's I've never been. It's on my list of places to go, but
It is far, but it's got so many different environments and places. You can go to the beach or the rainforest or the complete outback. So you've got it all here in Down Under.
I'll have to come visit sometime.
For
So
sure.
I wanted to ask you, what do you see in the future of your field? So you're studying now autoimmune diseases, cancer, a lot of infectious viruses. What do you think? Do you think we'll be able to cure some of these, or what do you see in the future?
Because I guess when I started this research, it was a very, very small field, and even though it is quite small now, the work that we're doing is really becoming more and more recognized, and we're really making some great advances with all the drugs we're identifying and these key proteins we're identifying in these processes. So we're very optimistic that we are going to really lay down the foundations to some key therapeutic developments in the future, which will really help us to understand these the roles of cell death in these different disease processes.
Yeah, that's really cool. Yeah, I think apoptosis, if you were able to control it and obviously control the clearance mechanism, would be a huge advance in cancer treatments and things like that, because in this case you want to kill a large amount of cells and have them cleared from the body.
Yes,
The ways
exactly.
we're doing it now with chemotherapy is just brutal, body. So that would be really cool if you could
Exactly.
target, a small section or whatever the tumor, and just kill that.
Yeah.
That would be really cool. Nice.
And I guess it's such a reflection on the technology and advances in technology and science these days, when you can perform these huge drug screens and screen all these different compounds and quickly see these results, whether they can be used in these kind of settings and whether they're showing these effects or not. So with the way the technology is going, all of these things are becoming a lot more possible.
Really cool, really cool. Now, one more personal question: has knowledge that you've gained from your research change the way that you live or the way that you look at life? This is a more profound question.
Yeah, I don't really think about that. I think it definitely does, but probably not in ways that I consciously notice. But I guess it probably makes you more aware, and I guess it really makes you think more holistic. Because I think in science, it's not just black and white all the time, but you really need to think outside of the box. And I think being in science and research really changes the way you think and evaluate things in normal life. So that's definitely a good trait that comes out of being a scientist, I think.
Yeah, and being able to, not just say this causes this, to be able to be like, but why, and to think about it deeper.
Exactly. Yeah. Asking those but why is really, really
but
important.
Why?
If you
Exactly.
read something on Facebook, don't just read it, just think, that's interesting, I'm going to look into that and understand how that works.
Yeah, exactly. Interesting. Well, one thing that has changed my way, or I guess outlook, is that I've realized how fragile the human body is. It's really fragile, and it sometimes can be really scary. I'm always walking around the clinic or in the lab, and I'm hearing about all these brutal viruses, and it's just it's crazy. The world we live in is really crazy.
Yeah, for sure. And there's just so much that we don't understand. I think that's quite so mind-blowing that you look at all the research happening around the world at the moment, and there's just still so much we have no idea how it happens or why it happens. So that's really cool and exciting for the future, I think.
Yeah, that's the cool part about science, is you're always learning. That's one of the reasons why I decided to get into the field. Cool.
For sure.
So another question, any advice for younger people that are looking to get into science in general or into your field?
I definitely recommend trying to get as much work experience as you can in your earlier years. So as mentioned, when I was going through my undergrad, I really had no idea what I wanted to do. And it wasn't until
like most of us.
Yeah, exactly. And it wasn't until that I got some work experience in the lab that I realized that, oh yeah, research is actually really cool. So I really recommend people just applying for internships or work experience or anything like that, all those opportunities, just to really figure out what you like before you really commit to it. So, and there are so many opportunities out there as well. So, there's no harm in applying for things. It's like definitely one of my mottos is just, you got to be in it to win it. Apply for everything, and it's a step in the right direction.
It's a numbers game. That's what I always say. I like to say the shotgun approach, where you just shoot the shotgun and see what happens.
Yeah, exactly. Yeah. And the way I think about it, so I always apply for scholarships and awards. Whenever I can apply for it, I may as well do it. And that whole process of applying for things or having interviews or writing their CV, it's all good practice anyway. So you're never losing anything from, putting yourself out there.
Exactly. Cool. Well, that's great advice. I wanted to ask you, since you said you were interested in science communication, what do you think, besides what we talked about earlier, are some of the problems with science communication nowadays? Do you think it's the Facebook and these, fake news, or what do you think is the big issue nowadays?
Yeah, it's definitely a double-edged sword, I think, with social media these days, because I guess a good thing is that, scientists and educated people who have, fact-checked their things can go out there and share it with the world, and that's really good. However, you can also get the other side of things where people may not necessarily get peer-reviewed articles or check it or, make sure it's true before sharing it. And that can be a very dangerous way of spreading fake news or things that aren't necessarily correct. And there are a lot of people who, even myself scrolling through Facebook, you may just say, oh, that's a cool article, and just believe it without double checking.
Yeah, exactly. So, yeah. Well, I think another issue I've noticed is that the study will come out, and then a news article will be written about the study, and then a news article will be written about the news article that was written about the study, and then someone will talk about the news article that was talking about it. And every step of the way, it gets changed a little bit. It's like the old telephone game you used to play when you were a kid. It's like, so, small changes happen, and then all of a sudden you end up with, 100% eating cheese at night causes you to sleep bad or something like this.
Exactly. we had a classic case a few years ago in our lab where some of the researchers in our group found that a small protein from the leaf of tobacco plants could actually kill cancer cells. And so
that's dangerous.
you could imagine that as soon as the media got a hold of that, it was smoking cures cancer. And it's
Exactly.
just such a classic example of how media can just definitely take the science and then just give you the wrong truth from it.
Exactly. Well, yeah, this is a problem. That's part of our mission statement. Deliver it right from the people that are studying it. Heard from them.
Yeah,
Cool.
yeah.
Cool.
Yeah,
Well,
for
I'm
sure.
running out of questions for you. Do you have anything else you want to tell the public?
No, I just think be curious and get into science, and that's it, really. Have a crack, and then just you really enjoy what you do, and science is cool, and don't forget that.
Don't forget that. Cool. So if people want to contact you, I guess we met over Instagram, so I guess you
Yep,
want to give your
that's
Instagram a shout out or something like that. So if people want to
yeah. So
contact you, that might be the best way.
people, yeah, definitely have a look at my my Instagram, which is at some blonde scientist,
Some blonde
and yeah,
some random blonde
Not-
scientist. We don't know her.
yeah, that's exactly it.
Cool.
So, but Instagram is great. There are so many different amazing bloggers and scientists out there to look at and to see what it is like to be a scientist. So get out there and have a look, and it's definitely an encouraging thing to see others out there struggling with science or just doing awesome science as well.
Exactly. Cool. Well, we will put all that information in the show notes, and hopefully you get some listeners that want to talk to you about your research or ask you questions or so. That's really cool. Cool. Well, thank you for
Awesome.
coming on. It's been a pleasure. Have a nice night. I don't want to keep you up too late. You probably have to wake up early tomorrow.
No problem. Thank you so much for having me. It's been great to talk to you.
Thank you. Okay. Bye-bye. Take care.
Thank you very much.